The journey begins within the complex architecture of the male urogenital system. Here, millions of specialized cells are prepared for a singular high-stakes mission. At the moment of climax, a chemical cascade triggers a powerful muscular contraction. Propelled into the unknown, they must navigate the acidic environment of the cervix. Of the 300 million who start, only a few thousand will reach the fallopian tubes. Inside the tube, the survivors undergo capacitation, the final chemical wake-up call. Out of 300 million, only a few thousand will navigate the treacherous uterine cavity towards the fallopian tubes. The cervical mucus, a protective barrier, also acts as a selective filter, favoring the strongest. Within the fallopian tube, the survivors undergo capacitation, their final chemical wake-up call. Meanwhile, the ovulation cycle culminates with the release of a single precious oocyte. Fimbriae, finger-like projections, gently sweep the egg into the waiting embrace of the fallopian tube. The egg, viable for a mere 12 to 24 hours, patiently awaits its destiny. Finally, the egg and sperm meet. A cluster of sperm swarm around the oocyte, a frenzy of activity. The first hurdle, penetrating the cumulus cells, a sticky, sugary matrix surrounding the egg. Next, the zona pellucida, a thick glycoprotein shell, the egg's primary defense. Upon contact, the sperm's acrosome releases enzymes, dissolving a path through this robust barrier. One lucky sperm finally breaches the cell membrane, its nucleus slipping inside. Immediately, the egg initiates the cortical reaction, sealing itself off to prevent any further entry. With fusion complete, two sets of DNA collide, creating a unique, brand new blueprint for life. Within 24 hours, the first cleavage occurs. One cell becomes 2, then 4, then 8. These cells, called blastomeres, remain contained within the zona pellucida as they divide. By day 3, it forms a solid ball of 16 to 32 cells, resembling a tiny mulberry, the morula. The morula continues its journey, propelled by rhythmic contractions of the fallopian tube walls. By day 5, a fluid-filled cavity forms, transforming it into the blastocyst, ready for implantation. The blastocyst must now breach its shell and burrow into the uterine lining, the endometrium, to survive. This vital process, implantation, anchors the nascent life and initiates nutrient exchange. The trophoblast cells multiply rapidly, forming a syncytiotrophoblast that invades maternal tissue. Maternal blood vessels are eroded, creating lacuna, spaces that will fill with maternal blood. This early interface forms the foundation of the placenta, the lifeline between mother and embryo. Crucially, the inner cell mass differentiates, forming the epiblast and hypoblast, the bilaminar disc. Around week 3, gastrulation begins, a dramatic reorganization forming the 3 germ layers. The ectoderm, the outermost layer, will become the skin, brain, and nervous system. The mesoderm, the middle layer, gives rise to muscle, bone, blood, and the cardiovascular system. The endoderm, the innermost layer, forms the lining of the digestive and respiratory tracts. Concurrent with gastrulation, neurulation begins, laying the foundation for the central nervous system. Errors at this stage can have profound consequences, highlighting the precision required for development. By week 4, a true marvel, the primitive heart tube forms and begins its rhythmic contractions. Initially, it's a simple pump, but rapid looping and septation will soon create a 4-chambered heart. Blood, though initially nutrient poor, begins its circulation throughout the developing embryo. The umbilical cord develops, a conduit of life connecting the embryo directly to the placenta. Within the placenta, a vast network of chorionic villi maximizes the exchange surface area. Oxygen and nutrients pass from mother to embryo, while waste products are efficiently returned. By week 6, tiny limb buds appear, the first hints of arms and legs like fleshy paddles. The facial prominences emerge, slowly converging to form the features we recognize. The eyes, initially on the sides of the head, migrate towards the midline, gaining definition. Internal organs continue their intricate dance of formation. Liver, pancreas, and lungs all take shape. Cartilage models the future bones, gradually ossifying into the skeletal framework. By week 8, most major organ systems are established, and the embryo is now officially a fetus. As the 3rd trimester matures, the fetus secretes the vernix caseosa, a waxy biofilm that prevents amniotic maceration of the developing skin. Inside the cranium, rapid synaptogenesis forces the cortex to fold into sulci, maximizing the surface area for a staggering influx of neural computation. Labor begins with cervical remodeling. Collagen fibers reorganize, causing the dense mucosal tissue to thin and stretch, a process known as effacement. The myometrium, triggered by oxytocin, begins a synchronized wave of pressure. The birth canal provides a mechanical squeeze, clearing fetal lung fluid, priming the intercostal muscles and lungs for the coming gas exchange. The first breath is a violent expansion. Negative pressure snaps the alveoli open. While the foramen oval in the heart snaps shut, rerouting the circulation. As the vertex emerges, the lumpy, distended mucosal tissues reach their limit of elasticity. Symbiosis begins with a tactile signal. The infant's suckling stimulates mechanoreceptors in the areola, sending an afferent surge to the hypothalamus. This triggers the posterior pituitary to release oxytocin, the conductor of the letdown reflex. Oxytocin travels through the bloodstream to the mammary glands, causing the myoepithelial cells surrounding the lumpy alveoli to contract. This propels colostrum, the liquid gold, through a network of wet, mucosal ducts toward the nipple. Colostrum is not just nutrition, it is a living tissue. It is packed with secretory IgA antibodies, leukocytes, and oligosaccharides that act as decoys for pathogens, providing the neonate with its first external immune system. As the colostrum enters the neonatal gut, it coats the lumpy, porous intestinal villi. This biological paint seals the leaky gut lining, preventing pathogens from crossing into the bloodstream while colonizing the microbiome. From the microscopic architecture of our cells to the monumental first breath, life is a masterpiece of biological precision. The journey of development continues every day. Thank you for witnessing the miracles of our beginning. If you're fascinated by the wonders of the human body, subscribe to Little Stork Corner and join our journey of scientific discovery.